In this picture, we are provided with an overview of the nervous system. Shown on the left of the figure is the picture of the presynaptic cell. Similarly, on the right side, we can see the postsynaptic cell. Shown in the diagram are parts of the neurons including dendrites, a nucleus, a cell body, an axon hillock, an axon, and a synaptic terminal. As you can see on the left side of the figure, the numerous branches coming out of the cell body of the neurons are called the dendrites, and the dendrites are where the presynaptic neuron receives the signal. Then, this signal is sent to the axon hillock, and the signal is transmitted through the axon. After this process, the signal eventually reaches the synaptic terminal, which causes the signal to be transmitted to the next synapse via neurotransmitters. The signal also causes neurotransmitters to be released through the membrane of the presynaptic cell, which thus binds to proteins embedded in the membrane of the postsynaptic cell. This signal often travels one direction through the neurons and travels very rapidly. This path can be shown by the red arrows in the diagram, ranging from the left side from the presynaptic cell, finishing off on the right side at the postsynaptic cell.
Here we see the important proteins used to transmit the signal through the axon of the neuron. An important part of the signal transmission is the concentration and voltage gradient. The concentration gradient is the force present that causes the diffusion of sodium and potassium ions from the high to low concentration. The voltage gradient is the force that causes the ions to travel to the side with their opposite charge. These forces are maintained by the sodium-potassium pump. The actual signal is transmitted with the help of sodium and potassium channels. They allow for the diffusion of sodium and potassium ions. More specifically, the sodium channels cause depolarization. Depolarization or reduction of the difference between the charges of the interior and exterior of the axon membrane is what transmits the signal. In this case, depolarization makes the interior of the membrane more positive. As this positive charge travels through the axon, it acts as the signal causing a conformational change in the axon terminal. In the membrane, there is a higher concentration of potassium ions and a lower concentration of sodium ions. This is very important to remember.
We can summarize the transmission of the signal in the axon in 5 simple steps. Step 1, the resting state. In this state, the gated Na+ and K+ channels are closed. Ungated channels in the sodium-potassium pump, not shown, maintain the resting potential. Step 2, depolarization. A stimulus opens the sodium channels. An inflow of sodium ions depolarizes the membrane. This brings it closer to the threshold, and if the threshold is reached, then the action potential is triggered. Step 3, the rising phase of the action potential. Depolarization opens most of the sodium channels. This leads to an influx of sodium and increases the positive charge of the interior through positive feedback depolarization. Step 4, falling phase of the action potential. Here most of the sodium channels inactivate, which stops sodium from entering the membrane. The potassium channels allow potassium ions to flow out of the membrane. This causes hyperpolarization or an increase in the difference between membrane potential. The interior of the axon becomes more negative. Step 5, undershoot. The sodium channels are closed, but potassium channels remain open. As this happens, the membrane potential decreases slightly under the resting potential, but then later returns back to normal resting potential. This graph details an action potential. The resting potential is shown by the flat line on the left of the graph. The resting membrane potential is around -60 to -80 millivolts. And then at this point, a few sodium channels open due to a stimulus, and the graph slowly starts to rise. Then, once it reaches the threshold, the graph suddenly increases at a rapid rate. The threshold is around -55 millivolts, which is shown by the dotted black line. Once the graph reaches the threshold, it increases at a rapid rate, because once the threshold is reached, many sodium ion channels are opened, which causes an Na influx, which causes a drastic increase in the overall charge. Then the graph curves and it begins to fall back down as most of the sodium channels are rendered inactive, which stops the inward flow of the sodium. Here the potassium channels begin opening, which causes potassium to flow out of the axon. This causes hyperpolarization, and the inside of the axon becomes more and more negative. At the end, some potassium channels stay open a little longer before eventually closing, which causes the membrane potential to dip slightly under resting potential before it eventually returns back to resting membrane potential. This is shown in the graph by a slight dip below the resting potential on the right side of the graph. Note that after Na pumps are activated, they can't activate again immediately. This waiting period is known as the refractory period, and the refractory period is what makes sure that the signal only travels in one direction from the cell body to the axon terminals.
This picture details how the action potential moves through the axon. Firstly, the action potential is generated as sodium flows into the cell inward across the membrane at one location. In the second step, depolarization of the action potential spreads to the neighboring region of the membrane, thus initiating the action potential there. To the left of this region, the membrane is depolarizing as potassium flows outwards. In the third and final step, the repolarization process is repeated in the next region of the membrane. In this way, the local currents of ions across the plasma membrane cause the action potential to be transmitted across the axon.
In this picture, we can observe myelin sheath in Schwann cells. The myelin sheath is a layer of electrical insulation that surrounds vertebrate axons. It is an adaptation that enables fast conduction of action potentials in narrow axons. Another advantage that myelin sheaths have is that they take up less space compared to thicker axons, which also increase conduction. They're produced by two types of glial cells, which are oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. This development occurs by the wrapping of axons in many layers of membrane, as myelin sheath provides electrical insulation for the axon. The myelin sheath's insulation causes the action potential to depolarize the electrical current to spread further inside of the axon. This brings more distant regions of the membrane to the threshold sooner. In myelinated axons, voltage-gated sodium channels are restricted to gaps in the myelin sheath, otherwise known as the nodes of Ranvier. These nodes serve as the point of contact between the axon membrane and the extracellular fluid. The arrows represent the action potential moving between the various nodes. As it reaches each new node, the action potential initiates itself. This mechanism is known as saltatory conduction, because the action potential is jumping from node to node along the axon. In this picture, we'll be going over synaptic terminals. Here's an overview of the synaptic terminal. This is the last point at which the signal from the axon reaches. When an action potential depolarizes the plasma membrane of the synaptic terminal, it opens voltage-gated calcium channels in the membrane. This causes an influx in calcium. The increased calcium concentration in the terminal causes synaptic vesicles to fuse with the presynaptic membrane. The vesicles release neurotransmitter into the synaptic cleft, which is the portion between the exterior of both synapses. The neurotransmitter binds to the receptor portion of the ligand-gated ion channels in the postsynaptic membrane. This opens the channels. In the synapse illustrated here, both sodium and potassium can diffuse through the channel. The neurotransmitter is released from the receptors and the channels close. Here, the synaptic transmission ends when the neurotransmitter diffuses out of the synaptic cleft, is taken up by a synaptic terminal or another cell, or is degraded by an enzyme. These neurotransmitters can either be inhibitory or excitatory. Inhibitory neurotransmitters cause hyperpolarization in the postsynaptic membrane. This prevents the action potential from happening, thus inhibiting the signal. On the other hand, excitatory neurotransmitters cause depolarization in the postsynaptic membrane, which allows the action potential to occur, thus transmitting the signal.